Okay, let's get started. So, a couple comments. So, a couple comments on the exam. Have a couple people that may take it yet. I'm kind of waiting to hear from them, so we're not going to go over any commonly missed questions. But overall, the mean was the 74, which is a little bit higher than the last exam. That's sort of in what we that teach this course kind of consider a reasonable average. So I did not curve it this time. Had one student get them all right, 100%. So this is just where we're sitting there. I often see this exam be higher overall, and it wasn't necessarily that this time. Some of you that had done better did worse on this one. So it was just kind of a mixed bag. So we have 2 exams left, right? We have exam 4, and then we have the final exam, which is weighted a little bit heavier. So if you're feeling crunched, you're not getting the grade that you want, come see me more often. I am not having people come see me during my office hours. I am giving additional office hours this week. If Friday morning is a time that you can come see me. It's usually pretty quiet up on the 3rd floor, Henry, at that time. You want to look over your exam. That's a good time to do it. The drop deadline has been extended till this Friday at 5 o'clock. So I've had a few of you come talk to me and want to crunch the numbers and just think about that a little bit. So remember what that means is after 5 o'clock on Friday, you can't easily just say, well, I'm doing bad, I want to drop this class. They usually won't let you do that unless you've had some emergency since the drop deadline. So you can take the class over again and replace the grade that you get here, but you can't just take it off your transcript. So if you again, go ahead and take it. So if you want to talk more about that, I'd be glad to do that. I don't typically, I'm not an advisor, like that's not one of my roles here. So, but I can point you in the right direction. All right, so we just kind of touched talking about introducing our big section that we're gonna spend some time on the next few weeks here, our immunology section. So we spent some time thinking about what's the difference between innate immunity and adaptive immunity in a kind of general way. We said that we were gonna approach this thinking about kind of our first line of defense, our second line of defense, and our third line of defense. And those are just artificial constructs. Like I'm not too worried that well, is this considered first line of defense or second line of defense? You should kind of have an idea of the difference between adaptive immunity and innate immunity. Right now, we're only talking about innate immunity. That just means the reactions and processes that take place are going to react the same way every time we're exposed to that particular pathogen. So there's not any learning that takes place or better response. For the most part, there's sort of a lot of exceptions as you learn more about this, but that's sort of the generally accepted thing. Okay, so if we look at our first line of defense, we're going to explore some mostly external barriers that we have, as well as some kind of chemical processes that would just kind of be considered our first line of defense. So a lot of these are things that might have other functions that we don't necessarily think about being an immune function. But the first thing that we think of is our skin. Our skin is really an impressive barrier. Sometimes we consider our skin kind of an organ all itself. And it really has some amazing adaptations so that skin is not a common portal of entry. Okay, only when the skin is breached somehow do we typically see that it can be a good portal of entry. Okay, so the skin is composed of many different layers. Okay, so let's see. Whoop. So it's many layers. The outer layers are going to be composed of dead cells. Okay, and those dead cells are continually falling off. We might say sloughed off. That's a process we also can refer to as desquamation. Okay, that just means those outer layers of cells are continually falling off. So that's important because, as we talked about in the process of infection, pathogens typically have to adhere to their target cells before they can invade deeper. So if those cells are constantly being lost, the ones they adhere to, right, they're not going to be able to establish an infection. Okay. And when you see this picture of our a scanning electron micrograph of our outer layer of cells, right, they look dead. That's no surprise. So have you ever thought about why we don't have viral skin infections, right? Like we might get rashes from a virus, and that's due to the work of the virus inside our body that shows that. But we don't get like bacterial skin infections like we get staph or strep infections on our skin. We don't get viral infections because those cells are dead on the outside. And remember, viruses need living cells to reproduce. So that makes sense. As we move deeper into the deeper layers of cells, what are we going to find there? We find lots of dendritic cells. And we're going to talk more about what dendritic cells are in a minute, but they're phagocytic cells that are patrolling around, looking for pathogens. We have sweat glands. So here I brought in this picture from the last chapter that just kind of a cross section of our skin. Here's just a picture of the sweat glands. Sweat is very salty, and we know that microbes, bacteria in particular, don't like to be in hypertonic mediums. They're going to dehydrate, they're going to lose water. Okay, so that salty environment is going to be inhibitory to bacteria in particular. Sweat also contains antimicrobial peptides. So I'm not gonna speak too specifically about that. There's just proteins there that work to inhibit microbes, as well as lysozyme is found in sweat. So remember, lysozyme, that's an enzyme that splits the two sugar molecules in peptidoglycan. So it acts to weaken bacterial cell walls. We also, in the deeper layers of our skin, have sebaceous glands. Okay, and that's what's pictured right here, that little kind of blue mass there. So that secretes sebum. Okay, and sebum acts to lower the pH of the skin. Okay, so skin, very inhospitable environment. It's a salty place. There's antimicrobial peptides. The pH is low. Just kind of an inhospitable place. Now, of course, sebum is important. The whole reason that we secrete that is more so to keep the skin pliable. When our cells secrete too much sebum, then we have trouble with our skin being oily, and it can block the pores up and contribute to acne. But if we get kind of the just right amounts, then that's works just right. Okay, so if we think about the defenses that we have in mucous membranes, we talked about how that is a more common portal of entry. We have lots of defenses at mucous membranes. They're just a little bit more easily breached by pathogens. And of course, our mucous membranes are the ones that line any opening to the outside. So our respiratory tract, our digestive tract, reproductive tracts. We have a very thin layer of epithelial cells, usually only a single layer of cells. They are tightly packed. And like I said, cells are alive, they're not dead like on our skin, but they are continually being sloughed off. So we do still have some desquamation taking place, even though it's kind of a thinner layer of cells. Those cells are continually being replaced to try to keep pathogens from adhering. Oh, I did say that right there. Okay. On these mucous membranes, we have cells that secrete mucus. And I just learned this like not that long ago, that when you use mucus as a noun, you spell it like this, M-U-C-U-S. When you use it as an adjective, as in like describing membranes, then you spell it with an O. Okay? English is kind of dumb sometimes, but I didn't spell that wrong. How do we produce that mucus on our membranes? We have a certain kind of cell, they're called goblet cells. Right, so this was the best picture that I could find of this. This happens to be a picture of cells in our respiratory tract. Okay, so you have a little bit slightly different kind of mucus produced depending on where in the body you're talking about. But from what I've read, all the goblet cells in your body produce about 1 liter of mucus per day, which seems like an astonishing amount. So I'm not talking about saliva, I'm talking about mucus. So, I mean, when I get allergies, I can kind of believe that that could happen because my nose runs all the time, but that seems like an astonishing amount. And that helps just move pathogens off those membranes and keep them from establishing themselves. We also, especially, particularly in the respiratory tract, or I should say only in the respiratory tract, we also have a different kind of cell called ciliated columnar cells, which have cilia attached. Okay, so of course that cilia acts to kind of help move pathogens. You know, we breathe in maybe some dust or some kind of pathogen, virus, bacteria, and that cilia action can help move it back out of the respiratory tract. If you're a smoker, then your cilia don't move quite like they should, and in some cases we lose the ability to produce cilia. The smoke can kind of damage those ciliated columnar cells. In general, smokers are more susceptible to respiratory infections because of that. There's evidence that smoking will dampen other kinds of immune responses as well. So if you need another reason to stop smoking, there's a good one. Also on mucous membranes, there's some antimicrobial peptides, things like lysozyme, some of the things that we see in skin as well. There's also going to be some dendritic cells circulating. Again, we'll talk more about those in a minute. There's a certain kind of antibody that's secreted onto mucous membranes. So we'll talk more about that when we get to our adaptive immunity question.
When people stop smoking, does the cilia like grow back or will
That's a great question. So does cilia grow back or is it permanently damaged if somebody stops smoking? I do not know the answer to that. Does anybody know the answer for that? I'll have to do a little research. It's a great question. I know they say that when you stop, like it's always a good thing to stop, like, but I don't know exactly the reason why. Okay. Other defenses. We talked a little bit a lecture or two ago about the role of our normal microbiota. Sometimes I catch myself, the term that we used to use was normal flora, and that's easier to say, and sometimes I want to say that term. We're supposed to say normal microbiota because flora makes it sound like it's a plant. Okay? So bacteria aren't plants, obviously, so I try to say normal microbiota. We know that our resident microbes play a pretty big role in helping with our overall immune response. There's a few ways that we can kind of understand that to some extent. We know that our residents contribute to this microbial antagonism. So what I mean by that is that our resident microbes are going to outcompete most of the time invading pathogens. Okay, so on our skin, we have a lot of resident microbes already, Staph epidermidis, for example. Any new pathogen that might be wanting to cause an infection is going to first have to compete with that Staph epidermidis for space, for nutrients, things like that, before it will be able to establish an infection. So we don't really understand very well why our immune system tolerates all this normal microbiota. It seems like there's some line that has to be crossed before our immune system will attack it. But we do know that our normal microbiota plays a really important role to stimulate some aspects of our immunity. Okay, so by our second line of defense, I mean mostly like the action of our white blood cells, for instance. And they've done some really interesting studies that help us understand that a little bit. They've been able to raise mice in an azenic environment. So that's kind of hard to tell what this is actually a picture of, but there's some little mouse cages in there, like, you know, a bunch of them, 10 or 12 maybe. And these were mice that were born in a sterile environment. They sterilized the bedding in there, they sterilized the food, they make sure that the air is sterile, and they raise these mice in sterile conditions. So essentially, they can say that these mice don't have any normal microbiota. It seems like it's fraught with difficulties to maintain those conditions. But what have they noticed about these mice that don't have normal microbiota? They don't grow very fast, and they don't ever get very big. So they're kind of spindly looking, scrawny animals. And if they do take a mouse out of that environment or infect it with the specific microbe, they find that those mice have very little immune response to it. So a pathogen that normally wouldn't kill a mouse almost always does if it doesn't have a normal microbiota. So we think that our normal microbiota, again, kind of keeps our other immune cells kind of stimulated or primed or ready to go. And we talked about before how just overall our residents kind of, they help us with a lot of aspects of our health, which we are learning more about all the time. We get some vitamin B5 from our resident microbes. That's a vitamin that is necessary for glucose metabolism. We get a little bit of their extra folic acid that they make, which we talked about is important to make DNA, as well as some amino acids as well. We get a precursor of vitamin K from our microbes. Vitamin K is essential for clotting. So again, that's a huge area of interest that I think is really going to take off over the next number of years, understanding which microbes need to be present in our bodies to make us most healthy. And you know, maybe someday we'll be able to ingest those microbes in pill form and we'll be healthier. So, okay. And then lastly, like I alluded to, there are lots of bodily functions that we don't think about them as contributing to our immunity, but they definitely have that as a role, maybe a secondary role. And not to memorize these are just some examples, things in our digestive system that, again, are important for us to digest food primarily, but also if you think about saliva, of course, we need that to wash our food down, into our digestive tract. But there's some antimicrobial peptides in saliva, things like lysozyme, which we keep referring to. It's most important to brush your teeth right before you go to bed, because overnight when you're sleeping, you don't produce as much saliva. And so if there are a high number of bacteria in your mouth before you go to sleep, okay, they're more likely to be successful in, adhering to your teeth and starting to break down that enamel. And people also that don't produce as much saliva as they should, there's some autoimmune conditions where that can happen. They have a lot harder problem with tooth decay. So, okay, saliva has some antimicrobial function in our mouth. Of course, stomach acid, low pH, as low as like pH 2, helps us digest our food, but it's also a pretty tough barrier for pathogens to survive passing through the stomach. Oftentimes they don't survive that, and then they don't make us ill even when we ingest them. One hallmark that we'll see here when we go through here is that a lot of different processes function to sequester iron. So iron is often a limiting nutrient for bacteria, especially. And if we have ways to kind of take up any available iron in our body, then that's going to make it harder for pathogens to gain a foothold. So we see that quite a bit. Anything where we're just, moving fluids through some, one of our tracts, it's going to inhibit the binding of pathogens. Just the function of moving urine out through our urinary tract, that decreases the chances that pathogens will be able to bind there. Again, any kind of secretion or flow, and again, you just keep seeing this iron binding proteins. Okay, so again, just some examples, not to memorize any of those, but just so you kind of understand, there's a lot of parts in your body that a lot of things that function to could be considered part of our immune response. All right, so moving forward, still talking about responses that are considered part of innate immunity. We're going to look first at the function of some cells. That might be the part of our talk here that's the most familiar to you. If you've talked about those different cells and their functions, maybe in anatomy at some point. There's some different proteins that are really helpful to complement or help other aspects of our immunity. We're going to take a look at some specific signaling molecules kind of briefly. Also want to talk about inflammation and fever and their role in our innate immunity. So that will probably get us through today. We'll see if we get to all that. Okay, so let's talk about the function of these cells that we find primarily in our blood. Sometimes they can leave our bloodstream and go out into our tissues or be found in our lymphatic fluid as well. How do we name these cells? Where do they come from? They're going to start out with some stem cell. Oftentimes they're going to start out in the bone marrow. Sometimes it's from stem cells in the liver or spleen. Okay, but then that stem cell is going to differentiate in a process that is known as hematopoiesis. Okay, so again, we could define that. That's kind of just the process by which the body manufactures blood cells, and they're going to differentiate into more specific kinds of cells. So we've got some kind of precursor cells that we would label erythroid, myeloid, or lymphoid, and kind of precursor cells, and then those differentiate into the different kinds of cells. So erythroid cells are going to differentiate into red blood cells, right? And we're not really going to talk about those anymore because they don't really have an immune function. One aspect of red blood cells that we will return to is that they originally had a nucleus, but then they lose their ability to have a nucleus. Yes. Oh, so, oh, this one. Oh, okay. So this is similar to the one that I posted right very begin with. So if you go back to, I'll come back to it in a second, but if you go back to this slide right here, it's almost the same thing. I phrased some of this a little bit differently, but so that's why I didn't reproduce it again. It's been a little bit since we had it. Okay, so yeah, so that just kind of a repeat of that. Okay, but thanks, I should have mentioned that. Okay, so again, red blood cells, not really immune function. They are not nucleated, and that what we're going to mention that later on. There is some things, some different receptors that are different, whether the cell has a nucleus or doesn't have a nucleus. So we'll revisit that. And then if we look at that myeloid precursor tissue, that can differentiate into platelets. Again, we're not really gonna, we do think that platelets have some immune function. Their primary function, of course, is to clot the blood, right? So we're not going to really explore that anymore in our just short discussion here. What we are interested here in the next cells here, the next three, we've got our basophils and our neutrophils and our eosinophils. And these are all, you can see the little kind of specks in the cells there. We refer to those all as granulocytes because they have these little granules in them. Okay, now you don't have to learn how to identify those cells just by looking at them. If you want to learn to do that, take a good histology course. So don't worry about that. But we're going to talk about the function of each of those granulocytes. And an additional type of cell here that comes from that myeloid tissue or myeloid cells are monocytes. When they're in the bloodstream, those cells are called monocytes, but when they leave the bloodstream, they're going to differentiate into either macrophages or dendritic cells. Okay, so again, we'll talk more about their function. And then lastly, this lymphoid tissue gives rise to lymphocytes. And there are three kinds of lymphocytes: B cells, T cells, and natural killer cells. We often refer to those as NK cells. Okay, so we're going to spend a lot of time talking about B cells and T cells, not so much NK cells. We mentioned them a couple times, but the monocytes and the lymphocytes, okay, these are considered agranulocytes. So A meaning without granules. And of course, if you take all of these together, all the granulocytes and agranulocytes, okay, those are all known as leukocytes, otherwise known as white blood cells. Okay. So we are most interested in the function of all those white blood cells that circulate in the blood, and they leave the blood at times as well. Okay, so we're going to look at all of those in particular here and talk about their function. Okay, so let's start out with neutrophils, because and we're going to talk about all three of the granulocytes. The neutrophils, they make up about somewhere 50 to 60% of the total white blood cells in circulation. Okay, so they are definitely by far the most common granulocyte, and they even will outnumber, like, say, the lymphocytes and the monocytes in the bloodstream. They have another name. They are also called polymorphonuclear leukocytes. Okay, you may have heard the term PMNs. Okay, that's what that stands for. They call them that because they have this, if you look over here at our picture, they have this many-lobed nuclei. Okay, so polymorphonuclear, many-lobed nuclei. Now, if you look back at this page, you can see our basophils and our eosinophils. They have many lobed nuclei as well, but because the neutrophils so far outnumber basophils and eosinophils, oftentimes when we say PMNs, we're really mostly talking about neutrophils. So if you use that term, like really specifically, a polymorphonucleocyte is either an eosinophil, a basophil, or a neutrophil. But again, there's so many more neutrophils in our body that we're usually kind of just referring to that. So those two terms are usually used synonymously. They are highly motile. And in fact, we often refer to them as our first responders. So they're often the first cell that's going to get to a site of injury or infection. How do they get there? They are moving by amoeboid movement. So they follow chemical signals. They're not just going there randomly. They chemotax. They follow chemical signals and move by amoeboid motion once they leave the bloodstream. And they are pretty short-lived. They're going to live in the blood somewhere between hours up to about 5 days. And once they leave the blood and move into the tissues, usually only a matter of hours. Okay, so when I say they're short-lived, yes, days at most, sometimes less than that. So that's not really a problem. When we talk about the function of them, I think you'll understand that that's not a huge problem. They're just continually replaced. If we need more, we make more. There's other cells that live a lot longer than that, other different kinds of cells. Okay. All right, so how do neutrophils act? Three ways. Probably the first two are a lot more common, but they are phagocytic cells, and we'll talk more about that process of phagocytosis shortly. They also act on pathogens by degranulating. Okay, so we'll talk a little bit about the kinds of chemicals that are in those granules, kind of similar to the kinds of chemicals that we find in our lysosomes. Things like digestive enzymes and also kind of inflammatory mediators. They will talk more about that. They also, this was described less than 20 years ago, that they can form nets. So that stands for neutrophil extracellular traps. Okay, and I think it was just about 10 years ago the people that discovered this got the Nobel Prize. So with these neutrophil extracellular traps, we think that the neutrophils have a way to kind of chew up their own DNA and expunge it outside of their bodies. If you've ever like done a mini prep of DNA in the lab, that DNA is really kind of sticky. So that DNA has some other kind of antimicrobial chemicals, peptides associated with it. And it just kind of throws itself on a pathogen with the hopes of immobilizing that pathogen. So in this picture, you don't even see the neutrophil because it's basically digested itself, more like a kamikaze pilot maybe. And this netting that you see is trapping that bacteria. That's a Klebsiella pneumoniae in a mouse lung. Okay, so the neutrophil has kind of exploded itself to trap it there, immobilize it, so it can't invade any deeper. And those proteins on there help to digest it. Other aspects of our immunity can come and maybe phagocytize it or take care of it. So that acts to kill the pathogen. All right, so here's another interesting picture. This one was just from Wikipedia. This is supposed to be a neutrophil. I don't think I posted this. It's a neutrophil phagocytizing some anthrax bacteria. In one way, this picture doesn't make sense to me because the neutrophil should be way, way, way bigger than a bacterium. But it's just sort of interesting, kind of like a, python ingesting a deer or something like that. How it's kind of sticking out down here. But okay, that's supposed to be a phagocytic cell. And then I just wanted to post this. This was just another graphic I found that just kind of sums up. This is how neutrophils work. They phagocytize pathogens. They degranulate that chemicals that act to bring about the death of the pathogen. Here they call it netosis instead of just nets. Again, neutrophil extracellular traps where they're just kind of expunging their DNA. There's some different enzymes here that would help to act on the pathogen as well. So just kind of a nice way to sum that up. All right, our other two types of granulocytes, I don't have as much to say about them. There are far fewer in the bloodstream. You can see eosinophils, only about 1 to 3% of our white blood cells in our bloodstream, and basophils even less. They both have kind of similar functions. They typically are just going to act by degranulation. I'll show you a picture of that in a second. They are generally not phagocytic, and they may act like that every once in a while, but that's not kind of their primary function. Okay, they don't do those nets. That's just a neutrophil thing. Eosinophils in particular are famous for targeting large eukaryotic pathogens, like helminth worms, so like flatworms, flukes. If you've got, if you do like a blood count, take a patient's blood and count the different kinds of blood cells that are in there. If you see a high number of eosinophils, then you suspect that that patient is either fighting some multicellular pathogen or there's some kind of allergic reaction going on. We see a role in increased inflammation and kind of inappropriate inflammation, allergic reactions for eosinophils, and to a lesser extent, basophils as well. So this is a picture that I wanted to show you. I didn't post this because I don't have a citation for where this came from. This picture showed up on my Pinterest feed a couple years ago. I was like, I don't know what algorithm they're using, but they have me pegged for sure on Pinterest. But it's such a cool picture. You can see all these eosinophils, they're chemotaxing, they're attracted to this parasitic fluke. Of course, it's much bigger than the eosinophils, and you can see how, you know, one cell or two degranulating is not going to have a big effect on something that's a lot larger than the cell. But when you get a whole bunch of them and they're all releasing their digestive granules, then you can see how that can bring about the demise of that pathogen. So it's kind of a cool picture to look at. Okay. Don't be afraid to jump in if you have questions or maybe try to clarify. Okay, so moving on to those different kinds of cells are agranulocytes. The ones without granules are monocytes and our lymphocytes. So we said that monocytes can differentiate into a couple different kinds of cells when they leave the bloodstream. Okay, there's not a ton of them in the bloodstream, but when they do differentiate, they live for a long time. So when they move out into the tissues, both the macrophages and the dendritic cells are quite long-lived. Macrophages are probably the most famous phagocytic cell. Sometimes you hear them referred to as professional phagocytes. Okay, so what they do, they number one are just always circulating throughout the body, and you could call them garbage collectors. They're going around, they're eating up dead and dying cells and dead tissues, just kind of cleaning things up for us. Okay, and they're also acting on pathogens. And like I said, we're going to walk through the steps of phagocytosis a little bit more carefully, but they do what we would refer to as processing and presenting antigens. So macrophages are really important, you know, to phagocytize that pathogen, and then when they present that pathogen to the lymphocytes, they're asking for help, basically, like, hey, this is what the innate system is dealing with. We need more help. We need something more specific to really capture this or get rid of this pathogen. So if you have a macrophage here, say this is a macrophage, this is somehow, this symbol is a little abbreviation for phage. That macrophage, say, is going to extend its pseudopods and ingest this pathogen. Okay, so inside there, that pathogen is usually going to be killed. In macrophages and in dendritic cells as well, they have a way to then advertise kind of on the surface of the cell what it was inside that phagolysosome that they were dealing with. So they chew it up and they're going to present parts of that, and that's what our B cell receptors and our T cell receptors are going to recognize and get them called into action and make a more specific response. Okay, so this little YouTube video talks about, it's just really short, it's just a little cartoon, kind of talks about the difference between macrophages and dendritic cells and kind of, you know, shows a little bit about that processing and presenting. It's kind of fun. So what else about macrophages? They communicate with lots of other cells by cytokine signals. Okay, eventually here we're going to talk a little bit more specifically about cytokines, mostly just kind of give you some examples of, hey, these are the kinds of molecules that we just refer to as cytokines. So they're always in constant communication with other cells. And like I said, they're fairly long-lived in terms of sometimes months. Okay, so when you compare a neutrophil and a macrophage, a neutrophil that may only live a few days, and macrophages that live a lot longer, we actually don't want neutrophils living very long because they are often acting to degranulate. And when they're releasing those granules, there are some inflammatory mediators in there that can cause damage to our cells as well. So we don't want neutrophils living a long time and keep degranulating over and over and over again. We want them to do it a little bit, and then if we need more, we make more. Macrophages don't have those granules. They don't act that way. We'll talk about how they know that something is worthy of being phagocytized, and then they only are going to harm, hopefully, the appropriate kinds of cells. Okay. Dendritic cells have a lot of the same function as macrophages. I talked about how they're plentiful, especially in the skin. They, again, they're phagocytic, and they process and present antigen. So we're going to talk more about what I mean by antigen. Maybe we'll get to that next time, but that's just the parts of pathogens that are going to elicit an immune response. Okay, so it might be, some protein on the outside of a bacterial cell that acts as an antigen, for example. Okay, and dendritic cells are really kind of funky shaped. That term dendrite, there's some root in there. I think the Latin, it means tree. So if you think about like the branches of trees, how it sticks out, nerve cells are kind of look like that as well. So they're kind of spindly like that. All right, and then just some basic information about our lymphocytes. We'll talk more about these later, but just for now, these are mostly going to be located in the lymph. Okay, so that makes sense. So that's why we call them lymphocytes. But there are a good number of them in the blood as well. So they're going back and forth between the blood and the lymph. And B cells. These are famous because when they get the right signals, they differentiate into plasma cells. And then those plasma cells are what produce antibodies. Okay, so antibodies are really important to fight lots of different kinds of infections. We say that our B cells are important in humoral immunity. I think maybe that's a term that you don't hear quite so often anymore. Humoral immunity refers, your body's humors are the fluids of your body. So we're thinking primarily in the bloodstream, all right? So those antibodies are circulating primarily in your bloodstream. They do leave your bloodstream, go out to tissues, go out to mucous membranes, wherever they're needed. But we think about our B cells mostly functioning in terms of making antibodies. T cells. Then we have several different kinds of T cells that we're going to talk about. Cytotoxic T cells. They are going to act on our infected cells. So say one of our cells that is infected with a virus, for example, a cytotoxic T cell, when it gets activated, will act to kill that cell so that it can slow down that virus replication. T helper cells, they really act to coordinate lots of aspects of our adaptive immunity. Our adaptive immunity has lots of checks and balances. There has to be kind of this signal and that signal and this other signal before cells act, so we can make sure that it's an appropriate action. And we talk about T cells mostly acting in cell-mediated immunity. Okay, so humoral immunity involves antibodies, and cell-mediated immunity is when our cytotoxic T cells are acting on infected cells. All right, so more about those later. We said that natural killer cells are also considered lymphocytes. They are primarily acting in our innate immunity. primarily acting on virally infected cells. So for a long time, we didn't really understand how they did that, how they understood that one of our cells was infected by a virus, because that's inside the cell. We understand that a little bit better, but that's really, our innate immunity doesn't have very good mechanisms to act on viruses. So that's kind of one of the only ways, through natural killer cells. Also a little bit through some particular cytokines that we'll talk about. But all right, so that's just a little introduction to our lymphocytes. Okay, I want to look a little bit more closely at this process of phagocytosis. Okay, we kind of went through that kind of briefly when we talked about anti-phagocytic factors a couple lectures ago. So when I talk about the process of phagocytosis, I'm referring to the kinds of cells that phagocytize. So that's primarily macrophages, neutrophils, and dendritic cells. These are our cells that primarily are going to do phagocytosis. Okay. And the really first step in this process is that those cells have to move towards where they're needed. All right, we call that chemotaxis, following some attractant. Okay, so they're going to follow the chemical signals released by the cells in that neighborhood already, and then it's going to call our phagocytic cells to that spot. Okay, so once they're there, they don't just start, gobbling up other cells, willy-nilly. They have to recognize that the cells are worthy of attack. Okay, so they need to recognize that those pathogens are foreign. And they do that with these receptors that they have all around the outside of their cell, and we call them toll-like receptors. And I like it when the names that we have for something is kind of intuitive. This is not intuitive because toll is a German word, and these were first discovered in fruit flies, and then we realized, oh, our cells have something that's similar to what we see in fruit flies, and so that's why we call it this toll protein on fruit flies. So anyway, these TLRs, these toll-like receptors, they're on the outside of our phagocytic cells, and what they do. Okay, so these cells right here, they recognize broad-based patterns that a lot of pathogens have. Okay, so they're not specific like antibodies are. There's about 10 different patterns that we've discovered so far that these toll-like receptors will recognize. So we call these broad-based patterns PAMPs, pathogen-associated molecular patterns. Okay, so some examples of these broad-based patterns that we might see on pathogens and not in our cells, things like lipopolysaccharide. That's an important component of that gram-negative outer membrane. Maybe peptidoglycan, right? Remember, we never have peptidoglycan anywhere in our cells. Something else like the protein flagellin. I don't know if there's one G or two there. That doesn't look right to me. But of course, our cells never have, well, not never. Sperm have flagella, but that's a common thing in bacterial cells and not something that is typically found in our cells. So those are just some examples of these pathogen-associated molecular patterns. And once the toll-like receptors recognize those patterns, then there's a signal that yes, this cell should be phagocytized. As well as I'm going to send out signals to other cells to call in other aspects of immunity. Things like complement proteins, which we'll talk about soon, or inflammation, which we'll talk more about soon, the role of those things. Okay, so It's always better, it's always easier to understand with a graphic. The book doesn't provide a graphic here. So here's an example here. Here's some different toll-like receptors. Okay, so here's, say this is a macrophage, for example. And it's got, we discovered, I think, about 10 toll-like receptors. There's a few other ones that have been discovered in mice. And you don't need to know, like, for example, toll-like receptor TLR4 recognizes LPS. You don't need to know that. But that's just an example. TLR5, flagellin, 2 L's, 1 G. I got that mixed up. And just kind of various membrane or wall components that we might find in yeast or gram-positive bacteria, peptidoglycan. Maybe there's one for mycolic acid. Okay, those are all things, again, that we never find in our cells. And some of our toll-like receptors are actually expressed on the endosome or the phagosome. So once we ingest it, then there's receptors that are expressed on that endosome that will detect things inside of that, say, bacterial cell once it gets chewed up a little bit. So things like double-stranded RNA, right? We never have double-stranded RNA in our cells. Now, we have single-stranded RNA and double-stranded DNA. They are a little bit different than humans if you have like a viral or bacterial DNA, because the methylation pattern is a little bit different. Different bases are methylated. So we have toll-like receptors that can, again, recognize different viral and bacterial nucleic acids. Okay, so obviously these toll-like receptors are not going to stimulate phagocytosis. That's already happened. But again, when those receptors are recognizes their PAMP, it's kind of like tripping a burglar alarm. Okay, like, oh, now lots of other, I'm going to send out signals to lots of other aspects of immunity to kind of help with this particular pathogen. Okay, so lots of other innate immune responses. Okay, so here's your graphic from your book that talks about the events of phagocytosis. So they've tried to kind of show you what it really looks like. There's a part of it's a scanning electron micrograph, and just the rest of it is drawn out so you understand a little bit better what you're looking at. But like we talked about, really the first step is that the cell has to get to where the site of infection is. It has to chemotax there. And then adherence, that's going to involve also recognition by the toll-like receptors that, yep, this is something that should be phagocytized. And adhering to that pathogen is not always an easy thing. We just talked about all these virulence factors that pathogens have to keep them from becoming phagocytized. Maybe they've got a capsule, for example. So we have things that can, fight against that as part of our immunity. We have things that we would call opsonins. Okay, an opsonin is any factor that's going to enhance phagocytosis, or in other words, make our white blood cells more likely to be able to phagocytize that pathogen. Okay, so some examples of opsonins, and we'll talk more about this soon. Okay, antibodies act as opsonin. They are specific, they bind to the bacterial cell, and then our phagocytes have receptors for those antibodies. They're like handles, so they can grab a hold of the pathogen. Another example that we may get to a little bit today are complement proteins. Okay, they bind to pathogens, and then the pseudopods on, say, the macrophage can use those as handles and is more likely to be able to adhere to that pathogen and ingest it. Okay, so we'll talk more about opsonins. So once that pathogen is ingested, we say that it's in a phagosome. That's just kind of a membrane-bound, endosome. Of course, then the lysosome has to bind to that phagosome, and then we consider it a phagolysosome. Lots of digestive enzymes in the lysosome. We're going to talk more about what's in there in just a second. 99 times out of 100, that pathogen will be killed by those digestive enzymes, and then it's going to be eliminated. Okay, we have mechanisms that actually pump hydrogen ions into that phagosome. Okay, so the pH is about 5 inside there. So that's another thing that kind of helps bring about the demise of whatever pathogen is going to be ingested. Okay, so what kind of things do we find within a lysosome? There's probably on the order of about 40 different enzymes. So we could spend a lot of time picking apart all that. Some examples of things that you find in there, lots of digestive enzymes. So, you know, some of these you talked about in labs, some of those extracellular enzymes that are proteases, enzymes that break down proteins, lipases, break down lipids. Lysozyme helps to tear apart the peptidoglycan cell wall. Enzymes that digest nucleic acids, and lots of others. Specifically, there's a couple of enzymes that are really kind of interesting to think about. Within our lysosomes, we make enzymes that are called respiratory burst enzymes. And that just means that we have a way to manufacture some of those toxic forms of oxygen that we've mentioned before, that, you know, normally are produced as part of normal aerobic respiration, but we have a way to make superoxide ion, hydrogen peroxide or peroxide ions inside our lysosomes then that act to, you know, as oxidizing agents, pull electrons off of molecules in that phagolysosome. And interestingly enough, neutrophils can make an enzyme called myeloperoxidase. Okay, and they can do a really cool thing. They can actually produce hypochlorite from hydrogen peroxide and chloride ions. Okay, what is hypochlorite? What's the familiar name for that? Bleach, right? You can actually make bleach in your neutrophils. Again, and that's just another tool to help your neutrophils be successful in fighting pathogens. There are genetic disorders where people are deficient and can't produce certain ones of these enzymes. So that just means that their phagocytic cells are not as successful in always getting rid of that pathogen. So that can be a real problem if your, you know, innate immunity is not functioning as it should. All right, and one more word about our phagocytic cells. They actually, especially macrophages, tend to have kind of 2 levels of activity. So they have their kind of normal level of function where they're going around and they are just, you know, cleaning up the garbage. Like I said, they're eating dead cells. But we know they have a higher level of activation. When they get the right cytokine signals, they become activated. And once they're activated, they just do everything better. All right. So a good example, Pac-Man, right? You know, when you play Pac-Man, he goes around and he eats the dots, right? But when he eats one of the big dots in the corner, then Pac-Man moves faster, he gets bigger, now he can even eat the ghosts. Okay, so same thing, exactly the same thing. Once a macrophage is activated, they get bigger, they are going to be able to phagocytize even more quickly, they make more lysosomal proteins, so they're more successful in killing the pathogen. They have to have a faster metabolism to keep up with that. They produce more cytokine signals to bring other cells to that spot to help them out. So basically, they're even better at killing invaders. There is a downside in that, you know, as a result of producing all these cytokines, they're going to have a higher level of inflammation and more likely to kind of be bad for you know, our tissues as well. But at least in the short time period, you know, it can be a really good thing. Okay. And this is kind of a fun thing. Let's see if I can get this to come up. This is, you think this would be easy to find on YouTube, but it's a time lapse. Oops.
Here, what you're looking at is a video of macrophages that have been given yeast to play with. And what you can see is the macrophages will move towards the yeast and then engulf it. So here one is moving directly towards it and then engulfs it. Here's another yeast that's here, which will eventually be attacked and eaten by another macrophage, shown right there. So in much the same way that Metchnikoff found, these invading microorganisms activated the macrophage, turned it on, increased its capacity for migration, its capacity for cytotoxic killing, and as shown here, its capacity for actually killing and eating the invading microorganisms. This is another movie showing that
they just do everything a little bit better. Okay, get back there. Okay, questions? All right, we are moving right along. Another major aspect of our innate immunity is our complement proteins. And I find that, I know that I was, you know, when I first started teaching this, I was a little bit less familiar with what this is. It's not as famous maybe as our white blood cells. But this is a really important aspect of our innate immunity. We call them complement proteins because they complement other aspects of immunity. We'll say of our innate immunity. Okay, so we are going to see how complement proteins can act and kill a pathogen. But most often, these proteins are just going to help out other aspects of our immunity. Okay, so this is a series of proteins present in our blood. They are made in the liver. They start out in our bloodstream, and then they can leave the bloodstream and go out into the tissues where it's necessary for them to fight infection. It's part of our innate system because, you know, when it's activated, it, you know, pretty much acts the same every time when it's activated. Again, there's no learning that takes place. And we call these cascade reactions because one complement protein acts on another, which acts on another, which then acts on another, which acts on, so it's kind of like, you know, the rock that starts rolling down the mountain at the top, and then it bumps into a lot of other rocks and can, you know, cause a big landslide. Okay, so what are the main functions of complements? Some of these little bits or complement fragments can act as opsonins. So we just talked about that. That means that they can aid, make phagocytosis more likely to happen. Some of the fragments can act to increase chemotaxis of cells. And inflammatory, or we'll just say inflammation. Okay, inflammation can be a really good thing and a necessary thing, kind of on the short term. Most of the time we think of inflammation as bad, but it's necessary, as we'll see. That will be our next big topic that we'll get to next time. Complements can form what we call MAC. Everything in immunology has an abbreviation. You have to kind of get used to that. That stands for a membrane attack complex, which can poke holes in pathogens. Okay, it would poke holes in our cells as well, except we have a lot of different proteins on the surface of our cells to down-regulate the effects of the complement that we produce. Okay, so a little overview of how this kind of begins. Begin thinking about this till we get to our more specific picture that the book gives you. But sometimes, and we're going to talk about there's different ways that this complement pathway can get activated. We'll talk about that at the end. But one of the ways is when we have an antibody that recognizes, when that first complement protein recognizes the antibody bound to an antigen. We'll talk more about what antibodies and antigens are later. But that first complement protein, so C1, the first complement protein, that's one of the ways it gets activated by recognizing that antibody antigen. And then it acts to split a couple of other proteins. Some of those bits form together and act on another protein, kind of act as an enzyme to split another protein, sort of so on and so forth. Some of these bits are important to increase inflammation, to increase chemotaxis of cells, to act as opsonins. And then some of those bits come together to form this membrane attack complex and poke holes in the membranes of other cells, which eventually you get enough of those and it causes the cell to lyse. Okay, so I know our time is winding down. Let's go through this quickly here, and then we'll kind of review it, begin with next time. But I've tried to be really clear about what I want you to understand, because this gets a little bit complicated with all the numbered complement proteins. So I'll refer you to my learning objectives on this. But that first C1, like I said, in this classical activation, it is activated by that antibody antigen. So here's a bacterial cell. Okay, that's kind of the antigen, we'll say. And here's some antibody that recognizes that. And the C1 protein gets activated, and that C1 splits these two complement proteins, C2 and C4. When say the C2 splits, we label the fragments as C2A and B. So the fragments are A and B. When C4 splits, we label the fragments C4A and C4B. So some of those C2 and C4 fragments then bind together and make the C4B, C2A. That's an important fragment because we have another name for that. We call that C3 convertase, because that acts then to split C3. And now, so now these are fragments now that are starting to have some function. C3A acts to increase the inflammation. Inflammation and chemotaxis kind of go hand in hand. So I'll just write inflammation. C3B actually acts as an opsonin. Okay, so that's the one thing I don't like about this picture. C3B, it actually binds up here. I'm going to draw it kind of big here so I can write on it. Okay, it binds back to that pathogen. Okay, you get a bunch of C3B on it, and again, those are like handles, so a phagocyte can get a hold of that a little bit easier. Some of the C3B binds back with the C3 convertase there and acts on C5 and splits C5. Now you've got C5A. It has the same effect. It triggers inflammation. C5B binds, so B for binds. A, you could think allergy, if that helps you think inflammation. The A's cause inflammation. And C5 binds up here, along with C6, C7, C8, and lots of C9s to make that membrane attack complex, which pokes holes, hopefully, in the walls of pathogens. Okay. So, and this is in your e-text. I would really encourage you, there's only a couple pages in your book that talks about complement. Read through that. That's gonna be helpful. One more thing to mention. So here's a picture of a bacterial cell that's been attacked by complement and has lots of these membrane attack complexes, all these little black dots. These are holes punched in the membrane. This is going to be act mostly on gram-negative bacteria. Gram-positives have that really thick layer of peptidoglycan, and those complement proteins just can't penetrate through that. But with gram-negatives, penetrate through the outer membrane, only a little bit of peptidoglycan, and clear through to the cytoplasmic membrane. So, especially acting on gram-negatives. Okay, I hope you have a good weekend, and come see me if you need to.